This lecture will cover the chapter on the prokaryotes. As I'm sure you already know, the prokaryotes consist of organisms that do not have membrane-bound organelles. So the the bacteria and the archaea are prokaryotes. So bacteria and archaea are the two domains of prokaryotes. And they're very, very tiny organisms. As we can see here in this image, there's a head of a pin at different levels of magnification. And when we zoom in close enough with a scanning electron microscope, we can see that the head of that pin is covered in rod-shaped bacteria. So around 3.5 or 3.6 billion years ago, we have the origin of life. This is in the middle of the Precambrian. We have abiogenesis occurring. Abiogenesis meaning life from non-life, or life, basically the boundary of chemistry into the field of biology. And this is thought to have begun with the formation of protocells. And protocells, like we see in this image here, are lipid bilayers that will form naturally if the phospholipids are present in the environment. So you don't have to build them, they form themselves. And they actually divide, and they are selectively permeable, and they have a few of the properties that you would expect a living cell to have. So if you have protocells forming and eventually incorporate genetic material into them, you could, theoretically, produce the first living cell. And there's a video about this. I highly recommend you check it out. The link is included on this page. But protocells are lipid bilayers that have some of the properties of life. Now, the basic structure of a prokaryote, whether you're an archaean or a bacterium, is the same. You have, first of all, what every cell has, a plasma membrane, or I'll just call it here a cell membrane. You also then will have in bacteria a cell wall to the outside of that membrane. Okay. Inside the cell, you're going to have DNA. And that DNA in this cell is going to be in the form of a large circular chromosome. It'll be all folded up, but we'll draw it as a sphere here. A large circular chromosome called a nucleoid. Remember, this is DNA. But there's also smaller rings of DNA inside of a bacterial cell, perhaps in archaeans as well, known as a plastid. Let me get this to work here. Plastid. So these little rings of DNA are called plastids. The large ring of DNA is called a nucleoid. Alright, forgive me if this video is a little choppy. I keep having folks come to my office door while I'm trying to record it. Okay, so we have a plastid, we have a nucleoid, and then we will also have inside the cell a ribosome. Well, more than one ribosome, but we'll have ribosomes in here. There we go. So ribosomes inside the cell. And of course, those are involved in protein production. So inside of the cell, you have your DNA, your lipid bilayer, so your cell membrane, cell wall. Another thing that I did not draw is called a capsule. It's composed of protein and sugars, and it surrounds the cell wall, and it helps to keep out toxins and perhaps things like antibiotics have a hard time getting through them. There's this structure called pili or singulars pilus that can be used to transfer plasmids from one bacterium to another. Some bacteria will swim around with flagella, and of course they all will also have ribosomes. Here's an image showing us a bacterium. We can see the cell wall, we can see the capsule. That capsule helps the bacterium to stick to this person's tonsil, allowing it to hold in place. So that cell wall is composed of a polymer of sugars and proteins called, well, polypeptides anyway, called peptidoglycan. And that's found in bacteria. And archaea have cell walls too, but they're not composed of peptidoglycan. They're composed of other substances. But the thing a cell wall does for these organisms, it helps to keep them from bursting in hypotonic environments. So osmotic pressure, water diffusing into the cell, causes the cell to gain water, but it won't swell up and burst because eventually the strength of the cell wall will prevent more water from diffusing into the cell. But in the same, with the same kind of logic, though, you can imagine why we've always used salt to preserve food, like beef jerky, let's say. You have food that's dried out and has a lot of salt on it. That will cause water inside the bacteria to diffuse out and dehydrate and kill the bacterium. So for those reasons, we've often used dehydrated or salty food as a way to prevent food from spoiling. When it comes to bacteria, which is the group we run into more often when we're talking about prokaryotes, there is a test you can do called gram staining, which will help you to determine what kind of bacteria you have. Now, I'm not talking about what species in this case, but bacteria fall into two major groups, the gram-positive and the gram-negative group. And you stain them to look at what type of bacterium you have. And the groups that will stain with this stain you call gram-positive. The group that will not you call gram-negative. And it's a technique used a lot in medical practices because gram-negative bacteria are often more resistant to antibiotics and pose more of a problem. So if we look at this diagram from the textbook, I'd like to point out that we have a gram-positive and a gram-negative bacterium represented. We can see here a gram-positive bacteria. I'd like you to look at the structure of that bacterium. We have a plasma membrane, and we have this cell wall composed of peptidoglycan outside of that plasma membrane. And this is the type that will actually stain fairly darkly using gram staining techniques. This peptidoglycan will absorb that stain and cause the bacteria to show up quite well. Now we can see kind of these purpley dots showing those. Now this bacterium over here is gram-negative. It has a little bit more complex structure. It has two layers of plasma membrane, so two bilayers, meaning so they have an extra level of selective permeability here, and their peptidoglycan layer is actually between, so their cell wall is between this layer of cell membranes. So we have the outer membrane, the peptidoglycan layer, and those together really are the cell wall, and then you have the normal plasma membrane. So there's more barriers for things here to get into the cell, which helps to make them more resistant to antibiotics. And this type of bacterium does not hold on to that violet stain as well. You see them here as pink, but you don't see any of the purple stain in the gram-negative bacteria. Now, any type of prokaryote can reproduce very rapidly because they divide by binary fission. So no mitosis, no meiosis involved. They simply copy that ring of DNA and split in half. And this allows them to adapt to a new environment very rapidly. But you'd have to at least ask yourself, how is this possible? They don't have sex, they don't have meiosis. How do they adapt to new situations? And I'd like to point out two things. First of all, they reproduce quickly, and there's tons of them in a small area, and there's going to be lots of mutations in that population. So even though mutations are rare, when you have billions upon billions of organisms in one spot, a few of them will have mutations, and those mutations give them the diversity they need to evolve. And I'd also like to point out that prokaryotes, we often think of them as primitive. They're really not primitive organisms. Remember, they have been evolving on this planet for 3.5 billion years. So the bacteria alive today are not necessarily the same as the first organism ever on Earth. They have been evolving on this planet for 3.5 billion years. They are not primitive organisms. Now, bacteria reproduce very rapidly and they have mutations. They also can change a little bit thanks to a process known as horizontal gene transfer. And this occurs with a pilus, that structure I talked about earlier. A sex pilus is basically like a tube that goes from one bacterium and stabs into another, allowing them to transfer DNA in the form of plasmids into that other bacterium. This is a one-way transfer, so the plasmid goes from one bacterium into the other. It's not sex, it's a transfer of a little bit of DNA. And this allows bacteria to pick up new traits. And this can happen between very distantly related bacteria. So you have this pilus form, you have a transfer of DNA from one bacterium into another. So like I said, mutations are rare in any organism. So how can a population of bacteria evolve quickly? Well, first of all, let's just talk about a population of a very common bacteria, E. coli. The probability of a mutation thanks to an insertion or deletion or a substitution of base pairs in any particular gene is rare. It's only 1 in 10 million times. So that seems really rare, right? 1 in 10 million times will a gene have a mutation. But in your intestine, there are 2 to 10 to the 10th power of these bacteria reproducing every day. That's a lot of bacteria. So even in your intestine, for any particular gene in that bacterium, there will be at least 2,000 mutations in that population. So you pick one gene and you look at all the bacteria in your stomach. That one gene will have over 2,000 mutations if you look at all of the bacteria produced in your intestine in one day. That's an incredible amount of diversity that can occur. So in your intestine, E. coli have 2,000 genes each. That means in your intestine there are over 9 million mutations a day in that bacterial population. So because of that, even though there's no sex going on, so they're not mixing up chromosomes, they have lots of mutations every single day, giving them lots of diversity. That means they can overcome antibiotics very rapidly. They can become resistant very rapidly, which is why we're having a crisis in antibiotics in this the current era we're living in. Okay, so another thing I want to mention, we talked a little bit about their biology. I want to mention that bacteria, or I should say prokaryotes in general, have really diverse metabolism, much more diverse than the eukaryotes. And we can look at different combinations of where they get their energy and where they get their carbon. So the carbon, of course, is what they use to build things, build new molecules, and the energy is what they use to drive those chemical reactions. So in human beings, we get our energy and our carbon from the food we eat, while plants get their energy from the sun and they get their carbon from carbon dioxide. So those are examples of eukaryotes. Let's look at the prokaryotes. One method is a system known as photoautotrophy. This is kind of like what plants do. Photoautotrophy means that there's light and self-feeding going on. So light and CO2 gas are utilized to create the energy for life. So CO2 gas and light energy are used to make molecules to keep the bacterium alive. That happens in cyanobacteria, for example. Chemoautotrophy is another form of autotrophy. This occurs perhaps in the ocean near thermal vents and things where carbon dioxide gas is used. but chemical energy is used to drive those reactions. So rather than light energy, chemical energy is used. Photoheterotrophy is when light energy is used to help break down organic material. So they bring in food to eat, but they break it down with the help of light energy. And chemoheterotrophy is kind of like what we do. Some bacteria do this as well. They bring in chemical energy and they break it down. They use some of it for their carbon source and they use some of it for their energy source. So chemoheterotrophy. So lots of diversity when it comes to bacteria in terms of how they make a living. And not just bacteria, but archaea as well, which allow them to live in lots of different places on the planet. So way back around, 3.5 or so billion years ago, we have the first prokaryotes showing up on the planet. And they were doing their thing. They were probably chemoheterotrophs for a while. That may have been one of the early life forms. But around 2.7 billion years ago, still in the Precambrian, we have photosynthesizers evolving and the oxygen revolution occurring. The oxygen revolution is a very important time in the planet's history. We start to change the atmosphere. And so we already know the atmosphere contains oxygen. And most of the oxygen that we breathe is of a biological origin. So without biology, there would be no oxygen to breathe. So if we look at this graph, we have atmospheric oxygen on a logarithmic scale on the y-axis, and we have time in billions of years here. So, 4 billion years ago, not a lot of oxygen. 3 billion years ago, not a lot of oxygen. But we get around 2.8, 2.7 billion years ago, suddenly photosynthesizers start activating, and we have this massive rise in the amount of oxygen in the atmosphere, all the way up to present day levels here. This oxygen revolution allowed the evolution of more complex organisms because oxygen was more available and organisms became more complex. Oxygen became an electron acceptor in the end of the transport chain, allowing for the evolution of modern cellular respiration. Another thing that bacteria do that's quite important is nitrogen fixation. Some of them live in the soil. Some of them live in symbiosis with plants, like we see in these root nodules, and they can convert atmospheric nitrogen to ammonia, which basically allows the plants to use atmospheric nitrogen then as fertilizer. So this is known as nitrogen fixation, a very important job that bacteria do. And a lot of scientists, including people in our department, are trying to figure out how to get other plants to live with these nitrogen-fixing bacteria, because the plants then, with the bacteria, produce their own fertilizer, which would mean countries where fertilizer is too expensive to afford could plant these plants, and the plants would fertilize their own soil. So lots of diversity in this group. We can see gram-negative bacteria here, cyanobacteria, which are the photosynthetic bacterias. Archaeans shown here. Notice archaea and eukaryotes share a more recent common ancestor with each other, forming a group. And the next chapter we'll talk about, we'll introduce the eukaryotes. So another thing that I want to mention about bacteria, as we already know, they cause disease, and we call these pathogenic prokaryotes. And pathogenic prokaryotes can be gram-positive or negative. Gram-negative just are often more resistant to antibiotics. And what antibiotics do is they often will either pop cell membranes or they'll shut off ribosomes. Those are the two of the major ways antibiotics shut down bacterial reproduction and bacterial growth. But bacteria in your body also are trying to fight you, basically. And this occurs in two ways. The presence of endo and exotoxins. So let's look at exotoxins at first. So exotoxins are toxins that bacteria secrete into their environment. So they do exocytosis, basically. They produce these toxins and they secrete them into the environment. The problem with exotoxins are that they will cause illness even if the bacterium is dead. So some food poisoning, for example, occurs with exotoxins. And even if you cook your food thoroughly, the exotoxins are still present and will cause illness after you cook the food. Okay, another distraction. I had to hit pause. All right, so exotoxins are secreted by an organism, and they stick around even if that organism's gone. So you have bacteria living in a piece of food that's in the fridge. Then you cook the food, you kill the bacteria, but the toxins remain. Endotoxins are toxins that are inside the bacterium, and when that bacterium dies and their cell walls pop, then that bacteria, that toxin will leak out into your system. So the bacterium has to be present, and a few of them might have died and leaked out their toxins, or they were killed and their toxins will leak out. And we already know bacteria cause lots of diseases, but remember, viral diseases and bacterial diseases are very different. You can treat most bacterial diseases with antibiotics, but not viral diseases. So a virus is basically, like we said, the first life form kind of crossed the barrier of life and chemistry. Viruses are still on the side of chemistry on that line. They are basically just proteins with some DNA for the most part. But bacteria cause tuberculosis, bacterial pneumonia, plague, syphilis, strep throat, etc., etc., etc. So there's lots of common bacterial illnesses, and I'm sure we've all had a couple of them. And just to point out this, some fun facts here before we close the lecture. There are millions of microbes on every square inch of your body, and over 1,000 species on your skin alone. So you are like a rainforest of diversity. And what's cool is your body is full of different habitats. So there are some bacteria that live on dry areas, some that live in sweaty areas because they like more moisture. And there are more, read this last one, there are more microbes in your intestine. So more microbes in your intestine than there are human cells in your body. So you are more microbe than you are human if you do a cell count. I hope that's at least a little bit disturbing for you. But the good news is a lot of these bacteria are not there to kill you. They're living in you and on you. You are their habitat. And some of them help us. They actually help keep out bad bacteria. Some of them help us make vitamins. They help us digest food. They help us do lots of things that keep us alive. And another really interesting thing, if we think of not just bacteria and archaea, the prokaryotes, but also like fungi and some of the other really tiny eukaryotes, we have members of every domain living in us and on us. So we are almost like a planet to these tiny little organisms. Every human being has a few fungi living on them, has some bacteria on them, has some archaea on them. We have every domain living in us or on us. And just hitting that point one more time, this is just a sampling of the bacterial diversity that a person can find in or on their bodies. And we do have some archaeans inside of our intestines as well. In fact, I've read that some archaeans may cause flatulence. I'm not sure if that's absolutely true or not, but we have them in us and on us. And if you look here, it shows you in between your toes, you have a certain diversity of bacteria. On your face and your nose, you'll have a certain diversity of bacteria. It's just pretty unbelievable how many different types of bacteria you can have living on you at any given time. So this chapter covers the little bit of the diversity, but we're focusing mostly on the general biology and the of the bacteria and the toxic effects on the human body and the different ways that these organisms get energy. And bacteria are very, very important organisms. You should read about it in your book. Read about some of the positive things bacteria do for us. One of the things they do is they're decomposers. They help to break down uh, dead organisms, releasing carbon and nitrogen back into their cycles. They also are used all the time for fermenting foods, for making antibiotics, etc., etc. An incredibly important group that is often ignored. I highly recommend you take microbiology before you graduate in order to get a better appreciation for this group of organisms.